Right now, we live in a world where scientists can clone entire animals. Crazy, right? But how does this work?
DNA, or deoxyribonucleic acid, is a molecule made up of nucleotides that complement and attract one another. This means that both strands stick together and they both make up the same genetic information. We make new cells from the old ones by growing and then dividing them. When a cell is ready to divide, its DNA must divide too, so there needs to be two identical copies of it, one for each cell. This is where DNA replication comes in. First, the DNA strands are pulled apart. Primase places an RNA primer on one end of the DNA. DNA polymerase builds new DNA on each strand, but it can only work if it starts building on an RNA primer. It only works in one direction. As the DNA is replicated, we find that the ends, or the telomeres, of DNA are slightly shortened since the primers have to attach to the ends of DNA, but new DNA cannot be built off of the ends. This is why telomeres are mostly protective, that is, they do not carry genes that make proteins, but contain nucleotides that can afford to be worn away over time. Many scientists theorize that this is a big molecular mechanism for dying of old age. When the DNA starts disintegrating, the cells of the body stop working. In 1996, Dolly the sheep was born. Why is this important? Dolly was the first mammal to be cloned from an adult somatic cell. Scientists took an udder cell from an adult sheep and reprogrammed it so that it would stop growing. Then they took an unfertilized egg cell from another sheep and extracted its nucleus. They inserted the first cell into the empty egg and shocked it so that the cells would fuse together and embryonic growth would begin. The egg was cultured for about a week until it started growing and its cells started dividing. Then it was implanted into a surrogate mother. Dolly lived a somewhat normal, comfortable sheep life at the Roslin Institute. She even gave birth to lambs, proving that a cloned animal can successfully reproduce. However, she only lived to be about 6 years old, whereas most sheep live to be about 11 or 12. Scientists believe that this is because Dolly's telomeres were too short. This happened because she was created with DNA that had telomeres that had already significantly disintegrated through the process of cell division.
Scientists have discovered a protein called telomerase, which is only found in reproductive cells and stem cells that extends telomeres.
However, we do not know how to use telomerase in our body cells, so unfortunately today's clones continue to lead relatively short lives.
We could theoretically clone animals that are on the brink of extinction and reintroduce them into the wild.
The reintroduction of species important to certain environments could protect or improve said environments, which has already been seen through the reintroduction of the Aldabra giant tortoises to the Galapagos. The tortoises brought numerous benefits to the island, such as boosting the production of endangered ebony trees, which rely on the tortoises for seed dispersal. It has also been shown that large herbivores help the environment by reducing carbon emissions. Many large herbivores are endangered or extinct because of us, so scientists believe that reintroducing animals such as elephants, sloths, and tapirs could actually reduce global warming alone.
While cloned animals may not be able to live as long, they are able to produce normal, viable offspring. Using this seemingly science fiction technology to reintroduce animals to populations where they are desperately needed could be the key to saving our planet from its impending doom.